Tacrolimus is a calcineurin inhibitor, and it's a very effective immunosuppression strategy as being a core immunosuppressant in preventing rejection in the first post-transplant year. And it's allowed us really to make kidney transplantation a mainstream therapy. However, there are some significant downsides to tacrolimus use long term. The first is that it's a vasoconstrictor and it's a profibrotic, if I can use that term. And so in the long term, it's associated with declines in kidney allograft function. So we will oftentimes lose transplants prematurely due to this progressive scarring process, which on a biopsy comes out as interstitial fibrosis and tubular atrophy, and which we think the calcineurin inhibitors, as well as a vasculopathy, so you get vascular changes as well. And so we believe that this process is something that really limits long-term allograft survival, which is our main challenge in kidney transplantation. The other side of it is that the drug itself has significant side effects or off-target effects. And many of those actually lead to premature death or contribute to premature death of the patients, namely hypertension, dyslipidemia, and what we call new onset diabetes. So patients who weren't diabetic before the transplant end up having diabetes after the transplant because of tacrolimus, which is a diabetogenic drug. So those are our major issues. The last thing, though, that I would want to highlight, and we haven't really talked about in the context of just biologics versus small molecules in general, is the fact that it's very difficult for patients to take small molecules. I'll use the word religiously for the rest of their lives. It's a very challenging thing to do. And so what oftentimes will happen is patients may have intolerances wherever, or they may have just, a bad luck where they can't take their drugs at a regular time interval. And so a drug that, provides therapeutic coverage in a biologic is much more attractive to patients in terms of long-term success. So those are sort of the big downsides of tacrolimus. Yeah, so tegoprubart is in a class of what we call co-stimulatory inhibitors. And so basically the T cells, which are the main cell that are the effector cell for rejection, you kind of, they basically have two switches. So you don't want them really turning on willy-nilly and causing, let's say, autoimmune disease. So these cells usually have what we call signal one, which is foreign antigen interacting with the T cell receptor, but then they need a second signal, which is this co-stimulatory pathway, of which tegoprubart is a monoclonal antibody against binding that. So it prevents the T cell from being activated in the first place. That's the first thing, without depleting it. But then because it binds on that co-stimulatory pathway, it also has effects on how T cells interact with their other neighbor cells, if you will. So things like B cells, T regulatory cells, it's much more favorable for the changes that occur in those cells in the setting of transplantation. So we believe ultimately it leads to a more favorable environment of more regulatory T cells, prevents B cell activation. And so overall, this is thought to be, from an immune perspective, a more healthy environment for the allograft to be in, if that makes sense. Yeah, so I think that the first thing and I was an investigator in the phase one study, so not really a big player in the phase two data. So it's nice to see this controlled data come out. And the first high level thing I'll say is the tegoprubart group did exactly as expected. It does have a higher rejection rate. That difference compared to tacrolimus was single digit, so it's 6%, which I would say is modest. Um, and then the other thing is it performed very well with regards to kidney function, which was the primary outcome of this study. So really spectacular achievement of kidney function here. What was a little bit surprising is I think a better than anticipated performance in the tacrolimus group, and I'm sure we can talk about that. But Tego, from my perspective, did as exactly as I thought it would do. I was anticipating a increase in rejection rate compared to tacrolimus. That difference was very modest, I would say. And the drug achieved a GFR of 69 mils per minute, which is really extraordinary level of kidney function. What I think is the difference here, just getting into the part that was a little bit surprising, is the fact that the tacrolimus group had a GFR of 66, and I think that's the characteristics of the types of kidneys that were included in the transplant. The other piece of this that is worth, I think, mentioning is that the side effect profile was not only as anticipated, it really made apparent the differences that we've already talked about between tacrolimus-based therapy and tegoprubart. The key differences here were really a huge difference in diabetes. We saw other factors that really matter to patients, which we didn't mention earlier on, including tremor, which was a significant difference. And then the other part of it, which is with any immunosuppressant, you're always worried about infections, and really there is no unique safety signals with Tego in terms of a big infectious risk issue. So those are all, I think, very favorable findings with this study. Yeah, sure. So just expanding on, the muted differences that we saw in the two groups with regards to eGFR at 1 year. Um, Again, I think it's that the tacrolimus group did surprisingly well, but when you look at that a little closer and you look in within subgroups, you see that this is really a low risk deceased donor transplant population. And so when the transplants that we're doing, today are not necessarily from a deceased donor perspective. This doesn't cover the spectrum. We're routinely accepting kidneys from older donors, and that's where you expect these differences in Tego versus tacrolimus to really manifest. So I think that this was a little bit of an anomaly in that you have a very low risk group of deceased donor transplants. That, of course, is to be seen in the context of a bigger trial. But I think a more real world, inclusive deceased donor population here would actually probably, really bring out those differences with regard to Tego compared to tacrolimus. I mean, I think that from an overall perspective, this is a green light for a phase 3 trial, I think is the first thing that's worth mentioning. I do think that it brings up some interesting things in here that we didn't necessarily anticipate. For example, we've long debated whether calcineurin inhibitors early on in the setting of really after the engraftment of the allograft, whether we get differences in delayed graft function. That's never really been interrogated in a trial, and it's interesting here that you do see a significant difference with the avoidance of a calcineurin inhibitor in the tegoprubart arm. Now, is that something that's going to pan out in a phase 3 study? I'm not sure, but it is an intriguing observation here. I think that the biggest thing that you want to see going forward for a phase 3 trial is the entire spectrum of kidneys that we transplant. You want to see how tegoprubart performs, because right now where I'm sitting, I'm looking at this data very favorably. I'm saying Tego, provided a very high level of GFR, but what I know from the other co-stimulatory trials, specifically the belatacept experiences, is that I anticipate that GFR to now be stable going forward in follow-up of these patients, whereas I don't anticipate that to be the case with the tacrolimus group. I expect attrition of the GFR over time. So the first thing I think is when I look at those GFR numbers, what is to be proven is are those going to be durable like we saw in the belatacept arm? And that makes sense conceptually to me based on all the immunological differences we talked about in Tego versus the tacrolimus arm. Yeah, I mean, I think the one thing that is important here is, I'm ex-president of the AST, and we performed a 10,000 patient survey, which showed that side effects of these drugs are a significant problem for patients. The one thing that we don't have in this study is patient-reported outcome measures. It would be great to see how the patients are feeling on this drug. Anecdotally, I can tell you that my patients that are on this drug in the phase 1 trial that are now continuing to take this drug now into the second year, they really like this regimen. So it would be great to see some patient-reported outcome measures into this, but at this point in time, I think that the results are very favorable for Tego at this point.